Welcome to BioWise with Achal. I am very happy that 2 units are done in 2 days and you are sincerely doing these units. Today we are going to talk about Unit 3. I hope you have subscribed the channel. If not, subscribe it right now and then proceed with the unit. Now in Unit 3 we study about human physiology and anatomy and nearly 50 marks, that is 45 to 55 marks is from this unit. Yes. Now this unit contains chapters like circulatory system, excretory system, nervous system, sense organ, endocrine system and reproductive system. So this unit is going to take little longer as compared to unit 1 and unit 2, which was only 45 minutes, but this unit is going to take little longer. So let us begin with the first chapter of this unit, that is circulatory system. Now what is circulation? The flow of fluid in your body is called circulation. What are the important organ systems responsible for circulation? The heart, the blood vessels, and the most important, blood. So let us take blood first and then we talk about the different parts of the circulatory. Proceed with the topic blood. Blood is the fluid present in our blood vessels which acts as a very important medium of transport of nutrients, respiratory gases and the waste, right? So what is the basic component of blood? Blood basically comprises of plasma and cells. Now it's around 55 to 60% of the blood is plasma, the fluid part of it, and remaining 40%, that is around 40 to 45% is your cells. Now what is plasma? Plasma is a fluid part of the blood. It contains around 90 to 92% of water, some proteins, and dissolved salts in it. Okay. You'll know that blood is a colloid. Colloid is what? It's a homogeneous appearing heterogeneous mixture. So here the cells are suspended in a fluid part that is called plasma. Now the cells part of the blood contains red blood cells, white blood cells and platelets. The scientific name red blood cells is called erythrocytes, leukocytes, that is for WBC, and platelets is thrombocytes. Now the importance of platelets is for blood coagulation and they do not have nucleus. White blood cells are nucleated cells and we have variety of white blood cells present in our body. We'll talk about it. First let us talk about the erythrocytes or the red blood cells. Now the red blood cells are biconcave cells flowing in your body which acts as a medium for the transport of oxygen. RBC contains a pigment called hemoglobin which combines with oxygen to form oxyhemoglobin and serves as a medium of transport of oxygen from lungs to the different tissues and organs. The blood which carries oxygen is called oxygenated blood and the blood devoid of oxygen or more of carbon dioxide is called deoxygenated blood. Now the hemoglobin can combine with carbon dioxide, but this combination is what to say, it's only 5% of carbon dioxide is transported in the form of carbaminohemoglobin. Rest 95% of carbon dioxide is transported as bicarbonate ions in the plasma. Okay. Now carbon monoxide, a poisonous gas, also has affinity for hemoglobin, 6 times more affinity than oxygen. So there is more amount of carbon monoxide in our atmosphere. We tend to in the atmosphere, it combines with our hemoglobin, making it unavailable for the oxygen transportation. Hence, we suffer from fatal symptoms. So, carbon monoxide poisoning is fatal for human beings. Right? Now, when you talk about erythrocytes or RBCs, RBCs do not contain nucleus, nor it contains mitochondria. The reason for not containing nucleus is it should have enough space for the transportation of oxygen and it contains no mitochondria because the oxygen utilization should not happen in the RBC. So, you have to remember that your RBCs do not contain nucleus and mitochondria. However, there are few organisms like camel and all, they do contain nucleated RBC. RBC loses its nucleus only when it gets matured and starts doing its job, that is carrying of oxygen. The lifespan of RBC is 120 days. Now, when you talk about WBCs are the soldiers of your body, helps in fighting infections. There are two types of WBCs, agranulocytes and granulocytes. As the name suggests, agranulocytes means granules are absent and granulocytes means granules are present. There are three types of granulocytes you can see. The one with numerous lobed nucleus is called neutrophils. Is called the neutrophil, numerous lobed nucleus. Okay, neutrophil. The one with bilobed nucleus is called basophils, and the one with the headphone shaped nucleus, that is you wear it in your ears, it is called eosinophils. So numerous lobed nucleus is neutrophil, bilobed nucleus is basophil, and headphone shaped nucleus is eosinophil. The agranulocytes means the granules are absent. There are two types of cells, monocytes with a kidney shaped nucleus and lymphocyte with a large nucleus. How will you differentiate? Lymphocyte has got the large nucleus and monocyte has got the kidney shaped nucleus, which is very small. So, largest WBC is monocyte, but the WBC with the largest nucleus is the lymphocyte. L for lymphocyte, L for large nucleus. So, this is all about the blood and its composition. Let us move to the blood coagulation. So, next we talk about blood coagulation. Before talking about the blood coagulation, as we have seen the different parts of the cell, here platelets or thrombocytes are very important for blood coagulation. Like WBC is for fighting infections. There is a unique feature of WBC that is called BC, which is called diapedesis. Now you have studied about amoeba, right? So WBC is also called to have an amoeboid structure, and they can actually move like amoeba, right? So when WBC can pass through the cell gap at the site of infection. This process of oozing out of WBC from the blood capillaries at the site of infection is called diapedesis. Now, WBC has reached the site of infection, so it will engulf the bacteria or the microorganism present. This process of engulfing the bacteria is called phagocytosis. So, diapedesis and phagocytosis, important words to know. Okay, coming back to thrombocytes or the platelets, which are very important for blood coagulation. Platelets releases an enzyme which is called thrombokinase when it is exposed to any kind of injury or injured cells or tissues. So let us quickly see the cascade of reactions that happens in the blood coagulation. Tissue and platelets, when there is an injured tissue, there is a wear and tear in the tissue, then in stress, the tissue along with the platelets release an enzyme called thrombokinase. Thrombokinase, in the presence of calcium ions and vitamin K, that is why we say, no, vitamin K is very important for our blood health. It helps in coagulation of blood. This is the reason. So, thrombokinase converts prothrombin into thrombin, which again acts as the catalyst for the conversion of fibrinogen. Fibrinogen is a soluble protein present in your blood. Since it is present in the soluble form, you don't have clots in your blood vessels. But when these reaction happens, this soluble protein changes into mesh-like protein or fiber-like protein, which is called fibrin. Now, fibrin forms a mesh-like structure and the platelets come and get trapped here. At the same time, the RBC will also come and get trapped here. So, this leads to the formation of clot, the blood clot, and the blood does not ooze out. Always remember, blood clot is possible only when the wound is little smaller. The big knife stabs and all, the blood clot will not be formed, because if the injury is too much, then that fibrin mesh cannot hold the blood flow. So, small wounds, blood clotting is very important. And there is a disease we have studied in genetics, hemophilia. Hemophilia is a disease in which the blood fails to clot. So, even the minute injuries in these people who have hemophilia, they can bleed to death. Okay. So, this is a very important mechanism for our body to prevent flow of blood or loss of blood from our system. This is called blood coagulation. I hope you understood blood coagulation. There are two terms that you should know. One is called plasma and the other one is called the serum. Okay, serum, these both are the fluid part of the blood. So the blood contains two components, as we have already studied, plasma and the cells, along with the proteins, that is the dissolved proteins in plasma, which helps in coagulation. Now after the blood clots, you have the formation of serum and the clot, and the clot contains all kinds of cells, right? So what is the difference between plasma and serum? Plasma minus clotting factors is called serum. Okay, so understand the difference between plasma and serum. Whenever a pathology person or anybody from the medical field, pathology department, they come to take our blood, they have got two vials. In one there is no salt present, but in another one there is a salt present. The salt is EDTA. Your work is to find out what is EDTA and put it in the comment section. Okay, now EDTA does not allow the blood to clot, so it remains in the soluble form. But if there is no EDTA present, the blood clots and the test can be done with the serum. Understood? Now there are few words, if you remember. Clot, the other term for the clot is also thrombus. Remember this, okay? Another name for clot is thrombus. And thrombokinase, the other name is factor 10. Factor 10 and thrombus, these are two new terms that you should know. Fine, so this is all about the blood coagulation. Now let us talk about blood group. We follow ABO blood group system, in which we have four types of blood group. One is A, B, AB and O. O basically is 0, not O, alphabet O, 0 because it does not contain any antigen. So on the basis of which antigen the blood contains, the name of the blood group is given on that basis. For example, A blood group contains A antigen, B blood group contains B antigen, AB blood group contains A and B antigen, and O blood group contains no antigen. What is antigen? Now if you talk about your RBC has got some kind of proteins present on its surface, which is called antigens. So if there is A antigen present, the blood group is said to be A. If the blood group contains B antigen, the blood group is said to be B. Okay. If the blood group contains both A and B antigen, it is said to be AB. And if it contains no antigen, then the blood group is said to be O. Understood? Now, depending if the blood group contains antigen, then there must be something which our body produces against these antigens. So, the person who has got blood group A produces B antibody. With B produces A antibody. With AB produces no antibody. That is the reason AB can take whatever comes to it. So AB is a universal recipient, and O contains both the antibodies. So O can receive blood only from O people, but can give blood to anyone. Reason is it does not have any antigen. Since it does not have any antigen, it cannot generate the antibody production in somebody else. So O is a universal donor and AB is a universal recipient. To be more precise, O positive, O negative, AB positive, AB negative, we must know the RH factor. Now, RH factor is a protein that we have inherited from our ancestors, rhesus monkey antigen. Okay, so the cells which contain this antigen is called positive, and the cells which do not contain this antigen is negative. So, if you suppose you have O negative blood group, O negative means no antigen present. Matlab this is nil, as well as there is no RH antigen present. So O negative becomes the universal donor. Okay, O negative becomes the universal donor. If you talk about AB positive means it has got A antigen, B antigen and RH antigen. Correct? So AB can receive any blood, because negative bloods can also be received, because negative bloods do not have Rh antigen, so it will not elicit the immune response. And AB positive receives all kind of blood, so it becomes the universal recipient. So AB positive is the universal recipient and O negative is the universal donor. In exam, if you don't remember the positive and negative, you can write only O and AB. Okay, but if you don't remember, don't write wrong things. Okay, but it's easy to remember. AB positive is universal recipient and O negative is universal donor. Now, a little bit integration with genetics. You see that the genotype of AB blood group is of two kind. This is homozygous A, this is heterozygous A. Since A is dominant, it will show the blood group A. This is homozygous B and this is heterozygous B. This is a combination in which both the dominant genes are together and both will express. This is called codominance. That is not in our genetic syllabus, but here both of them release their antigens. And this one is the recessive trait, that is O blood group. Fine. So, blood group has got 3 alleles instead of 2. Okay, like other characters, we have studied that there are 2 alleles, alternative forms of the gene. In case of blood group, we have got 3 alleles. Since it is more than 2, it is called multiple allelism. Okay, so what is a multiple allelism? Whenever a gene has got more than 2 alleles, it is called multiple allelism. This is just something important, as we can integrate with this topic with genetics. Fine. Now, Rh factor, as you have understood the positive blood, there is a disease which is called erythroblastosis fetalis. Erythroblastosis fetalis. Okay, as the name suggests, erythro, red blood cells, they rupture or die, and that leads to the death of the fetus. So, whenever a negative mother means the blood group is negative, carries a positive child, for the first pregnancy there is no issue, but for the next pregnancy the mother gets sensitized to the positive antigen of the baby's blood. Now, you have to remember that at any instant of time during the gestation period, the mother's blood does not mix with the fetus blood. But at the time of birth, there are chances that the mother's blood can get a tinge or a small amount of fetal blood, which makes the mother blood sensitive to the antigens present in the fetus blood. So, if a Rh positive child is delivered, the first delivery, no issue. For the second delivery, what happens? The mother starts producing antibodies against the positive Rh antigen and starts killing the fetal blood. You know that antibodies can transfer through placenta, right? So, it can go and kill the RBC of the baby, which leads to a disease, which leads to a condition called erythroblastosis fetalis. Okay, so this is all about blood group. Let us move ahead to the next topic, that is the blood vessels. Talked about the blood, the fluid which flows in the tube. What are those tubes? The blood vessels. What are the different types of blood vessels? There are 3 types: arteries, veins and capillaries. Arteries, veins and capillaries. When you see arteries, arteries have got smaller lumen and a wider tunica media. Tunica media contains muscles, basically the smooth muscles, and contains elastic fibers which provides it an ability to contract and relax. Now, why this is important? Because blood flows in pressure in the arteries, which exerts on the walls of the arteries. So, the walls of the arteries should be elastic enough to withstand that pressure. Okay. In case of veins, they have got a very huge lumen and they have got an unique structure which is valves. Okay, why valves are present in the vein? Because the blood does not flow in pressure in the veins. So, the backflow of the blood has to be prevented. That is the reason why valves are present in the veins. And the tunica media here is very thin, it does not have muscles and it is not elastic. Okay. This is the term used for the outermost layer, tunica externa, tunica media and tunica interna, and this yellow part is the lumen, okay, through which the blood flows. So in examination they generally ask you the difference between the arteries and the veins. So they can ask you the function as well as they can ask you the structural difference. When structural difference is asked, children, structure means what you can see in their structure formation. So you have to talk about the walls, that walls of the arteries are thicker as compared to the walls of the veins, and the veins contain valves but arteries do not contain valves. Is that clear? This is the structural difference. Functional difference, arteries generally carry oxygenated blood. Arteries generally carry oxygenated blood. Exception is there, which is pulmonary artery. Okay, we'll talk about it. Okay, pulmonary artery. And veins carry deoxygenated blood. Exception is pulmonary vein. You are going to mention the exceptions if the function is asked. Now, what is this then? This is a capillary. A capillary has got a single layer of cells, which is called endothelium or the epithelial layer cells. Squamous epithelium is present here. It helps in easy diffusion of gases, nutrients. That is why the capillaries exchange happens in the capillaries. So all the arteries, they form arterioles. Okay, these arterioles further divide into capillaries. These capillaries merge into venules, which finally goes into the veins. So you have arteries and veins and here capillaries. Fine. So this is how the structure is all about. So this is your blood vessels. Now let us talk about the most important part of your circulatory system, that is your heart. Now the human heart structure, I have drawn it to save your time, now comprises of the right auricle, the right ventricle, the left auricle and the left ventricle. Now there are 4 chambers in human heart. Generally with the evolution the number of chambers have increased. Okay, so earlier it was 1 pumping organ, then it was 2 chambered heart, then it was 3 chambered heart. Like reptiles and all, fish and all, they have 2 chambered heart. The crocodile has got 3 chambered heart, right? And human beings and mammals, they have got 4 chambered heart. Now increase in the chamber heart, increase in the number of chambers in the heart, increase in the number of chambers in the heart, increase the efficiency of the heart, so that the blood which is deoxygenated and oxygenated never mixes. So let us focus. In our syllabus we have the human heart only. So we have got 4 chambered heart, the 2 auricles and the 2 ventricles. Auricles pump the blood into the ventricles. Ventricles pump the blood to the entire body and the lungs for the process of oxygenation. There are valves present and there are valves prevents the backflow of blood. Now how the blood moves, right? In your body the deoxygenated blood moves from the, that is your superior vena cava and the inferior vena cava into the right auricle, into the right auricle. Right auricle pumps it to the right ventricle. From the right ventricle the blood moves into the pulmonary artery. That is why pulmonary artery carries deoxygenated blood. Now this goes to the lungs. In lungs, the process of oxygenation begins and the oxygenated blood comes back to the heart through the pulmonary vein and enters into the left auricle, from where it is pumped into the left ventricle. From the left ventricle, it is pumped into the aorta and to the entire body. Okay, so the blood vessel which pumps the blood to the entire body is called aorta, the largest artery. The largest vein is called the vena cava. Okay, and pulmonary vein brings the blood to the heart and pulmonary Artery takes the blood away from the heart. Arteries takes the blood away from the heart. Vein brings the blood into the heart. Arteries carry oxygenated blood except pulmonary artery, and veins carry deoxygenated blood except pulmonary vein. So let us understand about the circulation part. We have just circulation part. I have just changed this complex structure into a small box structure. Okay, so you have got the right auricle, the right ventricle, the left auricle, and the left ventricle. Correct? It now what happens? You have your body and you have lungs. Okay. Now what happens from the body? The deoxygenated blood goes into the right auricle. From the right auricle it goes to the right ventricle. From the right ventricle it goes to the lungs for the process of oxygenation. Lungs, the blood gets oxygenated and moves into the left auricle, then goes to the left ventricle, and the oxygenated blood moves to the body. So you can see there are two circulation that is happening. One is between the body and the heart, the other one is between the lungs and the heart. So the circulation of blood between the body and the heart is called systemic circulation. Systemic circulation. And the circulation of the blood from lungs to the heart and back into the lungs is called pulmonary circulation. And since there are two circulation happening, the entire process is called double circulation. Okay, double circulation. Let us name the blood vessels. The blood which comes to the right auricle comes through the vena cava. It can be superior vena cava and the inferior vena cava. The blood vessel which carries the deoxygenated blood from the heart to the lungs is called pulmonary artery. Then the blood vessel which brings the oxygenated blood into the heart is called pulmonary vein. Okay. The blood vessel which carries oxygenated blood from the heart to the entire body is called the aorta. Now these arteries or the aorta is subdivided into many arteries. Like the blood vessel which goes from the heart to the brain is called carotid artery. The blood vessel which carries oxygenated blood to your stomach and the digestive system is called mesenteric artery. To your kidney is renal artery. To your liver is hepatic artery. Right? The blood vessels which provide oxygenated blood to your heart is called coronary artery. So there are various arteries present. Different names are given on the basis of which organ it supplies the oxygenated blood. Fine. Now this is all the blood vessels, all the chambers of the heart and the double circulation. Now there are something called valves. Okay. Now valves, there is a valve present here, there is a valve present here, and there is a valve present here and here. The valves which are present in the right auricle and the left right ventricle is called the tricuspid valve. Tricuspid valve. Okay. And the valve which is present between the left auricle and the left ventricle is called the bicuspid valve. The other name is also called the mitral valve. Okay, these two are the valves present between the auricle and the ventricle. So they are called arterioventricular valve. Arterioventricular valve. Okay, these valves which are present between the ventricles and the arteries, that is aorta and the pulmonary artery, it is called the semilunar valves. So these two valves, the common name is semilunar valve because their structure is moon shaped. half moon shaped, semi-lunar valve. Okay, so this is called pulmonary semi-lunar valve, and this is called aortic semi-lunar valve. Is that clear? Now, these valves play a very important role in producing the sound of the heart, which is called lub and dup. Okay. So, how your heart functions. Now, if you consider your heart to be like this, the first is the arterial systole. Then there is a ventricular systole, and then there is a joint diastole. Arterial systole, ventricular systole, joint diastole. These three steps forms the cardiac cycle. Okay. So what you have? Arterial systole is what? Contraction. Then you have ventricular systole, and then you have got joint relaxation, that is joint diastole. Okay, now in these three steps, what happens when the arterial systole, it lasts only for 0.15 seconds, ventricular systole is 0.30 seconds, and joint diastole is about 0.40 seconds. Remember the table of 15, it's not 45, it's 40. So 15, 30, 40. So total 0.85 seconds ka cardiac cycle is there. Now in this cardiac cycle, what happens? First of all, just imagine a heart is full with blood. The first what happens, the arterial systole happens. So when the arterial pumps the blood into the ventricle, the blood moves to the ventricles. Now the ventricle has to pump the blood to the artery and the aorta. In that pumping, their blood should not go back into the auricles. It should go into only in the pulmonary artery and the aorta. So during this time, the arterial ventricular valve closes and produces the first sound, that is lup. Now what happens? The arterial systole, ventricular systole produces the lup sound. Then there is joint diastole. When the heart expands and the heart gets filled with the blood, but the filling of the blood starts, then there are chances that the blood which is there in the pulmonary artery and the aorta may come back into the ventricles. To prevent the backflow of blood from the pulmonary artery and the aorta into the ventricles, the semi-lunar valve closes during joint diastole, that produces the dup sound. So it makes a very important question for the board. Name the valves responsible for the production of lub and dup sound. The lub sound is produced due to the closure of arterioventricular valve, that is tricuspid and bicuspid valve, during ventricular systole, and dup sound is produced because of the closure of semi-lunar valves during the joint diastole. Is that clear? The lub sound. So we have talked about the chambers, the blood vessels, the lub and dup sound, the cardiac cycle. What is left? The impulse, or what gives your heart the rhythm to beat. Now there is a kind of special tissue present here, which is called SAN, sinoatrial node, which generate impulse for the contraction of the heart and the rhythm of the heart. So now our heart is basically a myocardial heart, where the muscles of the heart itself produces impulse for the beating of the heart. Now once this SAN node produces the impulse, it passes the impulse to the AV node, which is present here. This is called the AVN or AV node. Okay, it passes the impulse to the bundle of His, which again passes the impulse to the entire ventricles for the process of contraction, which is called Purkinje fibers. So SAN node, then AVN, and then you have bundle of His and Purkinje fibers. Remember the order, arrange in logical sequence. Question can be asked on this topic. So this is all about your heart, the beating of the heart, the sound of the heart, the flow of blood in and out of the heart, oxygenation, deoxygenation, chambers, and it is very, very important. Now you all know that the ventricles contract when it has to pump the blood into the artery and the aorta. This contraction phase is the systolic phase and the relaxation phase is called the diastolic phase. So systole happens in two steps, that is arterial systole and ventricular systole, and diastole is together. Okay, so this makes your cardiac cycle. Now you must have noticed that the left ventricle has got thicker walls as compared to the right ventricle. The reason is the left ventricle has to exert more pressure to pump the blood to the entire body, but the right ventricle has to pump the blood to an organ very close by, that is lungs. So this is, and even the arterial wall is thinner than the ventricular wall. Same reason, arteries have to pump the blood into the ventricles, but the ventricle has to pump the blood to the distant organs. Understood? So this is about your heart. There is something called blood pressure. Blood pressure is the pressure exerted on the walls of the arteries during the systole and the diastole. So the blood pressure is given by the systolic pressure, by the diastolic pressure. Systolic pressure by diastolic pressure. 120 mm of Hg by 80 mm of Hg. Okay, so systolic pressure is written up, this diastolic pressure is written down. 120 by 80 is considered to be normal. Now with the advancement in the pollution and other kind of stress factors, even 140 by 90 is considered as normal. But in exam, if the question is asked that what is the blood pressure of a human being, a healthy human being, it should be 120 by 80 mm of Hg. Is that clear? Then you have what is a pulse. You can feel your pulse here, right? This alternate expansion and relaxation of the radial artery because of this pumping of the blood. Every time the ventricles pump, there is a kind of wave produced in the arteries which is felt on the radial artery which is present in your wrist, in your feet, and also in your, that is, and also near your neck region. Okay, that is the pulse. The definition of pulse, definition of blood pressure. The instrument required for taking your blood pressure is sphygmomanometer. Please mind the spelling and write the correct thing. Stethoscope is to feel the heartbeat. Blood pressure is sphygmomanometer. There is a concept of lymph, like body fluids we have studied, right? Blood is one of the body fluid. Then you have tissue fluids or lymph. Lymph has another system, the lymphatic system. That is your drainage system. It is deals with the more of WBCs, fighting against infections, right? So it's a pale liquid. So whenever the blood goes to your capillaries, the blood cells remain in the capillaries and the fluid moves out, bathes the tissue, do all the exchange, and then gets collected back into the venules. If by any chance the fluid is not collected back, there is a tendency of accumulation of these fluid in your limbs that leads to a disease called edema. Okay, and that also happens if you have too much of salt concentration. The fluid osmoregulation of your body is hampered, so accumulation of fluid can happen in your blood, which is called edema. Now there is one more system, like you know that every organ is connected to a vein and an artery, but your stomach and your digestive system is having a blood vessel which comes from the aorta, which is called mesenteric artery. Now from the blood from your stomach and the intestine, there is a special vessel goes to the liver, because liver is an organ which plays a very significant role in detoxifying the material that you consume, and at the same time it also keeps an account of the nutrients that is reaching in your digestive system. So this portal system that is taking the nutrient from the digestive system to the liver is called the hepatic portal vein. Okay, so there is another portal system present in our bodies that is called hepatic portal system. Hepatic portal system is very important. Just after the meal, the blood vessel which has maximum amount of nutrient is your hepatic portal vein, right? Hepatic vein is the blood vessel which leaves your liver, deoxygenated blood, lots of urea, and it takes to your kidney through your heart, obviously. Fine. So this is hepatic vein is different, hepatic portal vein is different, hepatic artery is different. Is that clear? So let us have a quick review. Yes, so the last part of this chapter, this flow chart. I hope it's not complex. You already know the different blood vessels. Now the heart gives out the blood through aorta. The aorta then branches in many arteries, right? So the artery here I have forgotten. So the artery gets branched. So the blood vessel which goes to the head or the brain is the carotid artery. Okay, the carotid artery. Then the blood vessel which comes to the liver is called the hepatic artery. The blood vessel which comes to the stomach and the intestine is called the mesenteric artery. All names mesenteric artery. All names are not important, but it's better if you know, right? Then the blood vessel which goes to the kidney is called the renal artery. Okay, so these all arteries carry the oxygenated blood except for this, which is called the pulmonary artery, which carries the deoxygenated blood to the lungs, and the oxygenated blood comes to the heart by pulmonary vein. Now the blood vessel which brings the deoxygenated blood to the heart is called the vena cava, the superior vena cava and the inferior vena cava. Okay. This blood vessel which brings the deoxygenated blood from the liver is called the hepatic vein. This blood vessels, which is an interconnection between two organ system, is called the hepatic portal vein. All the nutrients and all will come to the liver first. The liver will access it. If excess of amino acids are there, urea is formed in the liver. Okay, and it is only filtered by the kidney. So urea laden blood will go to the heart, will travel through the aorta and then go to the kidney. So renal artery will have maximum amount of urea. Renal vein, this is renal vein. Renal vein will have the least amount of urea. Is that understood? So this is all about your circulatory system. I hope it's clear. Any doubt, put it in the comment section. Let's proceed to the next chapter, that is your excretory system. Now, excretory system is not that complex as your circulatory system, but it plays a very important role in your body in removal of the waste from your body, the nitrogenous waste. Now, urea is a basic component of the urine. They're in lower animals like fishes and all, they release ammonia directly into the water. So, they are ammonotelic. The birds, pigeon and all, you can see there, or lizards, you see that there is a white color discharge present on their stool. That is uric acid. And human beings and the mammals, they release urea, because urea is less toxic as compared to ammonia and uric acid and can be stored in your body for some period of time. Carbon dioxide is also an excretory waste that comes out through your lungs, and even your bile pigments are excretory waste that can be released in the fecal matter in form of bilirubin and biliverdin. Now, when we talk about excretion, any metabolic waste produced in our body, the major component of it is the nitrogenous waste. In us, the major component of urine is urea. You have to remember that. Urine contains water. It contains organic and inorganic substances. Organic is urea, uric acid, and inorganic component are the salts of potassium, KCl, and salt of sodium, NaCl. Remember this. So, organic component and inorganic component question is asked. Generally, the proteins are not released in the urine, so you cannot write creatinine and all. Slight amount of creatinine is present in urine, but excess of it leads to kidney malfunction. So, when organic and inorganic component of urine is asked, stick to urea and uric acid as organic component, KCl and NaCl as the inorganic component along with water. Now, the organ system comprises of two kidney. The right kidney is slightly lower than the left kidney because here you have the liver. That is why the right kidney is lower than the left kidney. Okay. The red part which brings the oxygenated blood is the renal artery, and the blue, that is renal vein. Now, when it comprises of kidney, ureter, urinary bladder and urethra, there is a circular sphincter present at the urethra which allows you to hold your urine for some period of time. Okay. In small children, when these muscles are not under the control, so they tend to pass out the urine, but you can control the outflow of the urine. Now, micturition is the process of expulsion of urine through urethra from the urinary bladder to the outside of the body. Please mention where, to the outside of the body. Functions of these organ systems are asked, like ureter, it carries urine from the kidney to the urinary bladder. Urinary bladder is a temporary storage of the urine. Urethra is a point of opening through which the urine is passed out. Kidney, which contains the structural and functional unit of filtration, that is nephrons. Now we can see this is the transverse section of kidney. It contains the renal capsule, renal cortex, renal medulla, renal pelvis, and the nephron is arranged in the cortex and in the medullary region. The entire pink portion is the renal medulla. Single units are called the renal pyramids. Okay. Now when you see here the nephron, the Bowman's capsule, this is the Bowman's capsule. Okay, this white part is the Bowman's capsule, and this one is the glomerulus. Together they are called the Malpighian body. Okay, they are called the Malpighian body or the Malpighian capsule. Always remember, the blood vessels which take the blood into the glomerulus is called the afferent arteriole A, and through which the blood moves out is the efferent arteriole E, exit. A for arrival, E for exit. So for arrival, the afferent artery has to have a wider mouth as compared to the efferent arteriole, because if more blood will enter and the less blood will move out, there will be a pressure developed in the glomerulus which helps in ultrafiltration. That is the first step of urine formation. Okay. Now this is the nephron. The Bowman's capsule, the glomerulus, the PCT, proximal convoluted tubule, and DCT, distal convoluted tubule, is present in the cortex. In the medulla, you have the loop of Henle and the collecting tubule. Please write it. All the point has to be considered, that which part of the nephron lies in the renal cortex, which part of the nephron lies in the medulla. So you have to write Bowman's capsule, glomerulus, proximal convoluted tubule, distal convoluted tubule is in the renal cortex, and the loop of Henle and the collecting tubule is present in the renal medulla. Now the steps of urine formation, the first step is ultrafiltration. Because of the difference in diameter of the afferent artery and the efferent arteriole, a pressure is developed in the glomerulus which helps in ultrafiltration of the urine. So everything gets filtered out, and there is a glomerulus filtrate which is produced, which moves into the proximal convoluted tubule. Now this glomerular filtrate has lots of important things which is not supposed to be thrown out in the urine. So the next step of urine formation is reabsorption. In reabsorption, all the important proteins, glucose, they are reabsorbed. Water is reabsorbed in the blood, allowing only urea and its component or water to move through the loop of Henle and go to the collecting tubule. Here also in the loop of Henle, the sodium reabsorption, chlorine reabsorption, active secretion means here the renal loop of Henle spends energy to actually take the material from the blood. That is, the blood, your body spends energy to throw out the substances like extra drugs, medicines. They cannot be filtered out. Because they are considered important by the body. So the body tends to spend energy and actively secrete these components into the urine to throw it out. That is called secretion. And finally, in distal convoluted tubule, there is a huge reabsorption of water takes place, because your body tries to maintain the concentration of water in your body. The important function of kidney is osmoregulation, and osmoregulation helps in retaining the content of water in your body. So if you are dehydrated, if you don't have enough water, then here reabsorption of water will be maximum. So you tend to pass out concentrated urine. If your body has enough water, then you tend to pass out dilute urine. This is done under the influence of a hormone which is called ADH, antidiuretic hormone. The deficiency of this hormone leads to a disease called diabetes insipidus, in which a person tends to urinate multiple times. That is called polyuria. So there are certain diseases that we must know which is related to your excretory system. One is glucoseuria. that is release of glucose in the urine. A person suffering from diabetes mellitus tends to release extra glucose in the urine, and the condition is called glucoseuria. Albuminuria, that means the albumin protein is not filtered out properly, is not reabsorbed properly by the kidneys, by the nephrons, so it is given out in the urine. The malfunctioning of kidney leads to the release of albumin in the urine, hence the condition is called albuminuria. Then you have hematuria. Hematuria is blood is passed out in urine. Okay, that is hematuria. Then you have polyuria. Multiple times urination is called polyuria. Fine. Then you have uremia. Uremia is accumulation of urea in the blood because of non-functional kidney. If your kidney is not able to filter out urea, the concentration of urea will increase in the blood, which will lead to a fatal condition that is called uremia. Okay, the blood becomes too acidic, toxic, so the person dies because of uremia. Is that clear? Then there are conditions like arthritis, gout, in which uric acid crystals gets deposited in between the joints, synovial fluid. So that causes friction between the two joints, and that leads to pain and arthritis. Okay. Then cholera. Cholera is a disease, that is a waterborne disease, in which Vibrio cholerae, the bacteria, causes the infection, and there is a release of toxins in the body. Fine. So the person passes out stool, watery stool. So there is a huge loss of water from the patient's body, and to conserve water, because of the ADH, the urine formation stops. So the person does not urinate, which leads to the accumulation of urea, no uremia. So that makes cholera fatal. Understood? Now, what if the kidney does not function? Then what will be done? So first, there is a temporary method by which is called dialysis or artificial kidney, in which the person's blood is filtered artificially in the hospitals using hemodialyzers, and till a person gets a perfect donor, then the person can go for kidney transplant. This is all about your excretory system. Now let us move to the nervous system. So the organ that is present in the skull, that is the brain, is the major part of your nervous system, and it is the boss of your body. So let us study the different parts of the brain. Now the brain has got, the major part is the cerebrum, and then you have cerebellum, and you have medulla oblongata. You can see this C-shaped, right? The hard substance which is present here is called corpus callosum. In reproductive system, we have something called corpus luteum. Okay, so corpus callosum, corpus luteum, difference can be asked. So corpus callosum is a hard part present in your brain, which separates your left hemisphere and the right hemisphere. And pons is a part of brain that controls the interaction between the left hemisphere and the right hemisphere. Okay. Now below the corpus callosum you have thalamus, and below thalamus is a hypothalamus, and there is small P-shaped gland which is called the pituitary gland. Okay, present in your brain. We are going to study about it in the endocrine system. Now when you talk about the layer which protects your brain, it is having 3 layers, and this is called meninges. Meninges is the covering of your brain. The membrane which covers your brain, it has got 3 parts: the dura mater, the arachnoid, and the pia mater. The dura mater is the outermost layer. Between the dura mater and the pia mater, there is a web-like structure present. That is why the name is given as arachnoid. Okay, arachnoid, and the last part is called the pia mater. These membranes are filled with a fluid which is called CSF, cerebrospinal fluid. This fluid is present in around your brain, and it continues till your spinal cord. Okay, and the meninges is the covering of your brain and the spinal cord. The brain has got the cerebrum, which has got many folds, which is called sulci and gyri, which increases the surface area of your brain to accommodate more of neurons. What is a neuron? Neuron is a structural and functional unit of the nervous system. Okay, now what is the function of cerebrum? Now when you talk about cerebrum, remember OTP. O, occipital lobe, T, temporal lobe, parietal lobe, and in front you have frontal lobe. Now what is the function of occipital lobe? Is for the vision and all the things related to what you see. So when you see something, it is your occipital lobe which comprehends what are you seeing. Temporal lobe is basically with your hearing and also related to your episodic memory. The frontal lobe is for intelligence, consciousness, and your parietal lobe is for storing the information and all the your experiences and all, it stores there. So this is your different parts of the cerebrum. Cerebellum is for control and coordination. An alcoholic person tends to walk in a very strange manner because the cerebellum is affected by alcohol. When alcohol is consumed, it goes directly to your brain and affects the cerebellum. The person loses the coordination. So the person could not walk properly. When we walk, we have a coordination between our hand, legs, muscles, right? That coordination is disturbed with the alcohol, so the person could not walk properly. Medulla oblongata is present here. This part of the brain deals with all involuntary actions of your body, whether it is your respiration, whether it's your heartbeat. When we are doing circulatory system, that time I told you that the heartbeat originates from your heart, but it can be regulated by your brain under the impact of hormones, also your nervous system. We'll talk about it, okay? Sympathetic nervous system. Now you know what are the different layers around the brain. What is the fluid present in the brain? This is the skull, which protects your brain from any kind of injury and mechanical shock. The different parts of the brain, that is cerebrum, cerebellum and medulla oblongata. The layers of the meninges, that is dura mater, arachnoid and pia mater, filled with cerebrospinal fluid. Okay. Now this is extended to your spinal cord. Always remember, when you talk about neuron, the cyton forms the gray matter. Okay, cyton forms the gray matter and axon forms the white matter. So our brain is a coconut, dark outside, white inside. So gray matter outside and white matter inside. Spinal cord, it is opposite. White matter outside, that is axons outside, and cyton inside. So always remember this. Okay, nervous system, when you talk about, there are two major components of the nervous system. One is central nervous system, that comprises of your brain, spinal cord, and the brain stem, which connects your brain and the spinal cord. PNS, that is peripheral nervous system, that is all that originates from your central nervous system. Now when you talk about the PNS, that is peripheral nervous system, you have something called somatic nervous system. It comprises of two types of nerves, which is called the cranial nerves and the spinal nerves. There are 12 pairs of cranial nerves and 31 pairs of the spinal nerves. Please learn the number correctly. You have to write in pairs. 12 pairs of cranial nerves, which are the nerves which originate from the brain, and 31 pairs of the spinal nerve, the nerves which originate from the spinal cord. Autonomous nervous system is what automatic, that is what is not under your control. So you have two components of the autonomous nervous system. One is called sympathetic nervous system, that means S for sympathetic, S for stress. So whenever you are under stress, your sympathetic nervous system activates. What it does, it increases your heartbeat, it increases your respiratory rate, it makes your pupil dilate, it stops your digestive system. So salivation stops, right? You feel dry. Then all the muscles of the body relax. So there's some cases you'll find even the sphincter in your urethra relaxes. So urine is passed out when you are too much scared. That all things are taken care of by the sympathetic nervous system. Which hormone it impacts? It impacts the release of adrenaline. So adrenaline is also called a stress hormone. So that is your sympathetic nervous system. Once the stress condition is over, the body comes back to its original state. The heartbeat normalizes, the breathing rate normalizes. That is done by the sympathetic nervous system. Let us focus on the neuron. Neuron is the structural and functional unit of the nervous system. It has got cyton, the dendrites, the axon, the myelin sheath, and the axon terminal. Whenever an impulse passes through the neuron, it leads to the release of neurotransmitters at the axon terminal. Example of a neurotransmitter is acetylcholine. Now this helps in the conduction of nerve impulse, leads to the transfer of impulse to the next neuron. So the gap between the axon terminal and the next dendron is called the synaptic cleft, and the process and the junction between the two neuron is called synapse. Remember synapses, that is crossing over? Here it is synapse. So what is synapse? Is a junction between the two neurons where the communication between the two neurons takes place. Now how does the transmission takes place? The most important ions responsible for transmission is sodium ion and potassium ion. When your neuron is in resting potential, that is when you are at rest, no impulse is generated, then no impulse matlab sodium out, Na out, no impulse, sodium out. So sodium is generally, sodium ions are present on outside of the neuron cell membrane. So once the impulse is generated, then the permeability of this axon increases for the sodium ion, and for every 3 sodium ion pumped in by the expenditure of energy, 2 potassium ion is given out. This is called sodium potassium pump, and it requires ATP. It's an active transport. Now when this exchange has taken place, the polarity of the membrane changes, and this is called the excited state or action potential. Okay, after that the impulse is over, and as soon as the impulse moves ahead, the permeability again changes back to normal, and sodium is again given out and potassium is taken in. So again it is come back to the normal state. So this is called the repolarized state, or it is again the resting potential. So resting potential is basically the polarized state. Once it gets depolarized because of the impact of the impulse, it is called action potential or depolarized state, and again it is repolarized to get back to the resting potential, and this is how the conduction of nerve impulse takes place. Any doubt, please put it in the comment section. Okay, there is a specific video on the every chapter present in the playlist. You can go through if you have not understood. Okay, this is a revision going on, so I am a little quick. I hope you understood the conduction of nerve impulse. Remember, what are the ions responsible? It is sodium and potassium. In normal state, sodium is out, potassium is in. When there is impulse, that is in the resting potential. It is also called polarized state. In normal condition, your neurons are polarized. Okay, after the impulse is generated or formed, then the depolarization happens. So action potential mein depolarization happens. After that again it has to come back to its normal condition. It is called repolarization. So resting potential polarized state, action potential depolarized, again resting potential repolarized. So polarization, depolarization, repolarization. This is the step of conduction of nerve impulse. On the basis of impulse your neurons get, there are 3 types of neurons. One is called the sensory neuron, which receives the impulse from the sense organs, from the outside world, which receives the stimulus, and it transfers it to the central nervous system. This is called your sensory neuron. Okay. Then in your CNS there are interneurons, which takes the information from the sensory neuron and gives it to the motor neuron. That is called the interneuron or association neuron. Motor neuron takes the information from the central nervous system and passes it to the effector muscle, which can be effector muscle or gland. So that is your receptor. So sensory neuron receives the stimulus from the receptor, passes it to the central nervous system. Central nervous system has interneuron or association neuron, which passes the information from the sensory neuron to the motor neuron. Motor neuron takes the impulse from the central nervous system to the muscles or the glands, which are effector, and brings response. So stimulus, central nervous system, response. Sensory neuron, interneuron, motor neuron. Depending upon the type of neurons a nerve contains. What is a nerve? Nerve is a bundle of axons of the neurons in a medullary sheath, that is packed in a medullary sheath. So if only sensory neurons are present, it will form a sensory nerve. If only motor neurons are present, it will form a motor nerve. If both sensory and motor neurons are present, it will form the mixed nerve. So I hope you understand the difference. Neuron, single unit, sensory, interneuron, motor neuron. nerve combined thing, sensory nerve, motor nerve and mixed nerve. Clear? Any doubt? Put it in the comment section. So this is about your nervous system. Now when we are talking about reflexes, that is how we respond to the stimulus. We have a very specific system which is called reflex action, that is taken care of by your spinal cord. So what is a reflex action? The reflex action is the immediate, involuntary, quick response to a stimulus. For example, you are driving a car and something jumps in front of your car. Without thinking, you will apply the brake. This is your reflex action. Somebody tries to hit you and you protect yourself. That is also a kind of reflex action. You place your feet on a sharp object and you jump. That is also a reflex action. You touch a hot object and remove your hand. That is also a reflex action. In all the reflex action, it is only your spinal cord which is involved. Your brain is not involved in the reflex action. Okay, this is your reflex actions. So the path of the reflex action is asked. Like what? The sensory, the sense organs. The sense organ receives the stimulus. So your path will be sense organ, receptor, that will go to your sensory neuron, will go to your spinal cord, from the spinal cord to the motor neuron, and then your muscles, which will help in retracting your hand. This is your reflex action. Then we talk about conditioned reflex or natural reflexes. Now, a child is scared from the height, that is a natural reflex. Conditioned reflexes is what we are trained to do. Tying our shoelace, cycling, that is all our conditioned reflex. Pavlov was a scientist who worked on conditioned reflex using dog and that is dog, and conditioning it to a situation where food was served to the dog with the ringing of the bell. So what he did, now whenever a dog sees the food, the natural reflex is to salivate. Now what Pavlov did, he gave food to the dog, at the same time he was ringing the bell. So the dog conditioned that whenever the bell will be rung, the dog will be provided with the food. So after some period of time, it was seen that even if food was not provided and the bell was rung, the dog started to salivate. So he proved that animals or human beings, they can be conditioned, they can be trained, right? So that is what he worked on, conditioned reflexes. So this is all about your nervous system. Let us move to the sense organs. Pick is sense organs. Sense organs we are going to study only about our eye and ear. Okay, other sense organs, skin we already had in 9th standard, we are not studying about it. Only eye and ear is important for your 10th standard boards. Now when you talk about an eye, this is the structure of the eye. It has got 3 layers, right? Sclera, choroid and retina. It has got lens, it has got iris, and the opening here is called the pupil. Okay, there are ciliary muscles which are present, which has certain ligaments called suspensory ligaments which holds the lens in place. Okay, which holds the lens in place. Then you have got this outer layer of the choroid which is transparent. This is called conjunctiva. Okay, this is called conjunctiva. And there is a fluid present here which is called aqueous humor, and the fluid present here which is called vitreous humor. So vitreous humor helps in maintaining the shape of the eyeball. Aqueous humor helps in the refraction of the light towards the lens. Okay, and at just a perpendicular to the principal axis of the lens, you have something called yellow spot. Okay, that is called maculae luteae, or you can call it fovea centralis, or you can call it simply yellow spot. This is the site of maximum vision, and here where you have the origin of the optic nerves, this place is called the blind spot. Any image formed here will not be perceived by your brain. Okay. Retina is a screen. So any image formed on a screen is a real image. We have studied in physics, right? So the image formed on the retina is always real and inverted. Always remember, the image formed on retina is real and inverted, which is then processed by your brain to make it seedha and you can see it, or make it upright and you can see it. Fine. So when you talk about the lens, whenever the object is at distance, the lens elongates to increase the radius of curvature in the focus so that the image is formed on the retina. In your physics, you know that the lens, you can move the lens so that the image is formed on the screen. But in case of your eye, the distance between the lens and the screen cannot be changed. So the lens changes its shape to accommodate the vision. Accommodation is far and near vision. And how does your lens accommodate your vision? Is your lens becomes elongated when it has to see a farther object, and it becomes short and round when it has to see the nearer object. This is called accommodation of your eye. Fine. There is one more thing which is called adaptation, that is light adaptation and dark adaptation. That is when you move from a dark place to a bright place, then your pupil constricts, right? For certain period of time you will not be able to see the things clearly. So that is a time of adaptation. When you move from a bright area to a dark place, the pupil which was constricted will now dilate. So again there is some fraction of seconds you will not be able to see things. Now when you switch off the light at night to sleep, you realize that the whole room is dark. But when you get up at midnight maybe for drinking water or any other purpose, you will realize that you can see the things around. This is what your adaptation is all about. Now next we have photoreceptor cells. You know that macula lutea fovea centralis yellow spot is the site of maximum vision because it has maximum concentration of cones. Cones are for color vision and rods are for the dark vision or raat ka vision. Okay. Now rods contain a pigment called rhodopsin and cones contain a pigment called idopsin. Please remember the name, rhodopsin and idopsin. Rhodopsin is very sensitive to bright light. So whenever you are out in the bright light, the pupil constricts and the rods bleaches. So what happens? You cannot see for certain period of time. When you move from the bright light to the dark, then the rod which was bleached will again regenerate, and that fraction of second is required for you to have a proper vision in the dark adaptation. Is that clear? So you understood about rods and cones. These are photoreceptors mainly concentrated at macula lutea. This is the site of maximum vision precision. That is, when you see at an object, you focus on one object, rest are all blurry, right? So the focused image is formed on the macula lutea or the yellow spot. Fine. This is the normal eye, right? When the image is formed on the retina. Always remember children, whenever you are drawing the diagram for the eye, you need to use pencil and please use arrow to draw the ray diagram. Okay. There are conditions which is called myopia. Myopia means short sightedness. That is, you can see the object which is close to you, but you cannot see the object which is far. Far objects are not seen. Hyperopia. That or hypermetropia is far sightedness. You can see the object which is far, but the nearer objects cannot be seen. Okay, nearer objects cannot be seen. Then you have astigmatism. Astigmatism is something where you have multiple images being formed on the retina because of the difference in thickness in the cornea. So you have multiple images. So some images are very distinct, some is not seen even, and some are blurred. Generally differentiating between different lines, horizontal and perpendicular, cannot be done by the people who have astigmatism. At the same time, there is something called presbyopia. Presbyopia is in the old age, people cannot see the objects that is very close to them because of the suspensory ligament or the ciliary muscles become loose and they cannot make the lens contract and relax accordingly. Then there is something called cataract. Cataract is your lens has to be transparent, right? So there are certain proteins which are present in vitreous humor which is in soluble form, but with the old age starts getting deposited on the lens, making the lens opaque. The opaque thing appears only on the periphery, so the people have halo vision. Slowly and steadily the entire lens becomes whitish and opaque and the person loses its vision. Okay, so this is your defects of the eye. Yes, so let us talk about the defect. Now in general case, what happens when a person is suffering from myopia, the image which is formed is in front of the retina. Okay, this is the normal myopia case. And case of hypermetropia, what happens, the image is formed beyond the retina. Okay, so here is the image, here is the object, here is the object, and here is the image. So this is the condition. In both the cases the person will not be able to see the things clearly. Now what happens, we need to know how it is corrected. So for correction, what is done in case of myopia, concave lens is used. Now you know that concave lens is a diverging lens. So what happens when the light falls on the concave lens, it diverges and then the image is formed on the retina. So the myopia is corrected. So in case of hypermetropia, what is happening? The image is formed beyond the retina. So here the lens used is a convex lens. You know that convex lens is a converging lens. So what it does, it just focuses the light little bit towards the center. Okay, and the image is formed on the retina. So the vision is corrected. So this is, drawing is very important. What are the things that you have to keep in mind when you are drawing? Use pencil for the drawing. Use arrow mark for the movement of light, the same way you do it in physics. So the arrow mark is very important, where the light is following, where the image is being formed, and label it correctly. The part of the eye can also be labeled, but you have to label the lens, the correction. Any question can be asked regarding this correction of myopia and hypermetropia. This is all about your eye. You have to draw the proper diagrams. Now what is left is the stereoscopic vision of yours, that is binocular vision. You have two eyes, two retina, two images are formed, but we see only one. That is done by our brain. So we have a 3D vision because of this binocular vision. Okay, then there is something called persistence of image. Generally in movies and all, what happens, 24 or more images are shown in a second time. So it appears as a moving object. Fine. Images are formed by a concrete photograph kind of structures. Each photograph is replaced by the next photograph in a very less amount of time. So you feel like continuous videos. Video is nothing but, because of persistence of image, you can see various photographs just compiled one after the other and moved with a very fast speed. So that is all about your eye. Let us move to ear. So let us quickly go through ear. First of all, what is the important function of ear? The major function of ear is hearing and balance. Okay, how balance? We'll talk about it. Hearing and balance. Okay, as we had photoreceptor cells, here in ear also we have the sonoreceptors or phonoreceptors, that is called the organ of Corti. Organ of Corti are the sensory cells responsible for hearing, present on the basilar membrane of the cochlea. We will come to know about it. First, understand the structure of the ear. The yellow part, what you can see, is the outer ear. Okay, outer ear. The middle part is the middle ear, that is the pink part is the middle ear, and this white part is the inner ear. Okay, now inner ear is responsible for hearing as well as for balance. How? Now what happens, the sound travels in the ear, right? The pinna, this is the outermost, here you wear your earrings. Okay, now this pinna is responsible for collecting the sound into the opening in your ear, which is the auditory canal. Now auditory canal receives the vibration. There is something called ceruminous gland which is present here, which releases earwax or cerumen or cerumen. Cerumen is nothing but earwax. Okay, earwax helps in dampening the vibrations and also trapping the dust and microorganisms if it enters in your ear. Now this is called the eardrum or the tympanic membrane. Tympanic membrane vibrates and transfers the vibration to these three bones, which are called ear ossicles. The collective term of these three bones is ear ossicles. The names are malleus, incus, and stapes. Malleus, incus, and stapes. Stapes is the smallest bone present in the human body. So malleus transfers the vibration to incus, incus to the stirrup or the stapes. The common name is hammer and will stirrup. Scientific name to be written in the exam: malleus, incus, and stapes. Fine, together they are called ear ossicles. Then in the middle ear you have Eustachian tube. Now Eustachian tube opens up in the throat. Okay, it opens up in the throat. That is why any infection that happens in the throat reaches your ear, or anything you put in your ear reaches your throat, like medicine is felt in the throat when it is put in the ear, or ear drops are felt in the throat when it is put in the ear. The reason is the Eustachian tube is connected to the throat. Now Eustachian tube in maintaining the pressure. So it equalizes the pressure on both the sides of the eardrum. That is why when you fly in the aeroplane or you go to a hilly area, you find that your ear is blocked, because there the pressure is low and inside the pressure is high. So you feel a outward pressure on your eardrum. Okay, even bombs and all, when they explode, it is seen that the people go deaf, eardrum burst. Why? Because of the bomb, the extremely low pressure is created outside. So from the inside, the air pressure burst opens the eardrum. Anyway, this is, then there are two windows. This is connected to the inner ear by oval window, and there is one more window called the round window, which has got thin membrane, which connects your middle ear to the inner ear. Now your inner ear has got these semicircular canals which are perpendicular to each other, three semicircular canals perpendicular to each other. These semicircular canals are responsible for your dynamic balance. Whenever dynamic balance question is asked, that is your spatial orientation. How when you are in motion or when you are moving, that is all taken care of by the semicircular canal, basically this part, which is the ampulla of the semicircular canal. So which part of the ear is responsible for the dynamic balance? The perfect answer will be ampulla of the semicircular canal. Only ampulla will not make any meaning, because ampulla is a swollen end. So ampulla of the semicircular canal is the right answer. This part is called vestibule. Okay, vestibule has got two components, utriculus and sacculus. Utriculus and sacculus are responsible for static balance. So there is a vestibular nerve which is connected to your brain, which helps in finding out whether you know balance or not. So this is your utriculus and sacculus, responsible for static balance. Ampulla of the semicircular canal responsible for dynamic balance. Then now these all things are filled with fluid. The fluid present in your ear is perilymph and endolymph. Perilymph and endolymph. Okay, these fluid is present in the inner ear. So all the vibrations here, these ear ossicles are amplifying the vibration, and the fluid present in the ear starts producing ripples. So the vibrations are basically converted into the ripples in the fluid, and then the brain senses what we are talking about, right? So this is the auditory cochlear nerve. Cochlear nerve. So these two nerves are actually combining to form the auditory nerve. So auditory nerve, vestibular nerve, cochlear nerve, and auditory nerve. This cochlea is containing three canals. The first one is the vestibular canal, which is close to the vestibule. Tympanic canal, which is close to the you can say tympanum, that is middle ear, and in between you have the median canal. The vestibular canal and the tympanic canal contains perilymph, and the median canal contains endolymph. In the median canal, there is a tectorial membrane and the basilar membrane. The basilar membrane has got the sensory cells, which is called the organ of Corti, responsible for hearing. So is that clear? This is all about your ear. Let us move to the next chapter, that is your endocrine system. So next topic is endocrine system. In endocrine system, we have only 4 glands in our syllabus, that is your pituitary gland, adrenal gland, pancreas and thyroid. Location of each gland, the function of each gland, hyposecretion and hypersecretion of each gland has to be known. Okay. Then metabolism, that is insulin and thyroid stimulating hormone ka feedback mechanism is important. Now let us proceed. Pituitary gland is a small P-shaped gland located at the base of the hypothalamus. You remember the diagram? Yes. So in that, the P-shaped gland present at the base of the hypothalamus brain, that is below the hypothalamus, is called the pituitary gland. It is also called the master gland because it controls and regulates the activity of the other glands. So pituitary releases 2 types of hormone, a general hormone and tropic hormones. Tropic hormones are those hormones which makes the other endocrine gland function. Now endocrine, exocrine, I hope the difference is clear. Endocrine are ductless glands, they release their secretions in the blood and it goes to the target site. Exocrine have ducts. Okay. Pancreas is a dual gland. It is endocrine as well as exocrine. I explain pancreas. When I'll explain pancreas, I'll tell you about it. Let us talk about pituitary first. So there are 2 lobes of pituitary. One is called the anterior lobe and one is the pituitary posterior lobe. The shape of the pituitary is like this. It is from the base of the thalamus you have in the brain, and then you have hypothalamus, and there you have a small P-shaped structure which is called pituitary. Pituitary releases growth hormone. Pituitary releases growth hormone, excess growth hormone. Pituitary releases growth hormone. Excess of growth hormone leads to gigantism in human beings. Less of it causes dwarfism. Now, if in adults, right, in adults you have acromegaly. A for adults, acromegaly. That is the limbs, the fingers, the jaw, the chin becomes large. So that is acromegaly. So hyposecretion of growth hormone is dwarfism. Hypersecretion is gigantism. In adults, it is acromegaly. Fine. Next one is prolactin. Prolactin is a tropic hormone that is responsible for secretion of milk after the childbirth. ACTH, all the full forms can be asked in the exam. Adrenocorticotropic hormone. It triggers the adrenal cortex. TSH, thyroid stimulating hormone. It triggers the thyroid gland. LH, luteinizing hormone. It is responsible for ovulation in females, right, on the 14th day. So luteinizing hormone again targets the ovary. Follicle stimulating hormone in formation of primary follicles for the production of sperm and the ovum. So again this is a tropic hormone. So next posterior lobe of pituitary releases 2 hormones. One is ADH. We have already studied about the function of ADH in the excretory system chapter. It helps in reabsorption of water from the distal convoluted tubule. Deficiency, hyposecretion of ADH causes diabetes insipidus. Okay, and oxytocin is for the uterine contraction. Oxytocin is the only hormone that is there for the positive feedback. That is, when oxytocin is released, more uterine contraction happen. More uterine contraction causes more oxytocin to be released. Okay, so positive feedback is shown by oxytocin. Next gland is adrenal. Adrenal gland is located on your kidney as a cap-shaped structure on both your kidneys. Now, adrenal gland has got 2 structures, outer cortex and inner medulla. The cortex has releases 3 types of hormones. The first one is mineralocorticoids, that is also called aldosterone. glucocorticoid, which is called cortisols, and sex corticoids, which are called androgens. Now, mineral corticoids are responsible for the regulation of sodium and potassium ions. Glucocorticoid helps in the metabolism of carbohydrate, proteins, and fats. And sex corticoid, androgens, helps in they actually produces hormones, sex hormones of both males and females. Over secretion may lead to premature sexual maturity. That is, the sexual maturity comes early in the children, those who have more release of androgens. Moreover, release of more androgen leads to a condition called adrenal virilism. Adrenal virilism is when a female gets a mustache and the beard because of the excess of androgens present. Then the next medulla middle part, they release a hormone which is called adrenaline. Adrenaline is also called a stress hormone. Abscisic acid is a stress hormone in plants. Adrenaline is a stress hormone in human beings and animals. Okay, so adrenaline is released by medulla. It is also called the fight, flight, and fright 3F hormone. Okay, responsible for managing your stress. So this is all about your adrenal gland. Coming to pancreas. Pancreas is basically, this is your stomach, okay? So this is your stomach, this is the duodenum. So the pancreas is present here as a leaf-like structure. So is as a leaf-like structure. So location of pancreas, pancreas is located in the loop of duodenum behind the stomach. You have to write this as a location of pancreas, loop of duodenum behind the stomach. Behind the stomach. No other answer will be accepted for the location of pancreas. Now, pancreas is a dual gland. It is endocrine as well as exocrine. The ducts of Echinus, which we have already studied in 9th standard, is the exocrine part of the pancreas. Pancreas has got small island-like structures which is called islets of Langerhans. There are different types of cells present in islets of Langerhans: the alpha cells, the beta cells, and the delta cells. Now, delta cell somatostatin is not there in our syllabus, but it is just the hormone which counteracts the growth hormone. So, somatotropin, that is your growth hormone, and somatostatin together regulates your height. Okay. Glucagon means glucose gone. कहाँ से gone? From the liver into the blood. Okay, so level of glucose in the blood increases because of glucagon. Insulin means in the cell. So the level of glucose decreases in the blood. So increase in insulin causes the lowering of the glucose level in the blood, that is hypoglycemia. Increase in insulin, decrease in insulin, that is diabetes mellitus, is a condition where insulin is less. Hyposecretion of insulin causes diabetes mellitus, which is hyperglycemia. Okay, now here in adrenal cortex, hypersecretion causes Cushing syndrome and hyposecretion causes Addison's disease. I forgot that to mention. Addison, A comes first, so it is hypo, and C comes later, Cushing's, so this is hyper. Okay, hyper secretion of adrenal cortex hormone, right? So this is your hyper secretion from the adrenal cortex causes Addison's disease. Hyposecretions from the adrenal cortex causes Addison's disease, and hypersecretion causes Cushing syndrome. In Cushing syndrome, there is hyperglycemia because the glucose metabolism is hampered. Okay, so this is coming back to the pancreas. Glucagon helps in the release of glucose in the blood by conversion of glycogen into glucose. Insulin helps in decrease in level of glucose in the blood by allowing the cells to take glucose. So they both work together to maintain a optimum glucose level in your blood, that is around 80 to 120 mg per 100 ml of blood. Last is thyroid. Thyroid hormone is thyroid gland is stimulated by TSH, thyroid stimulating hormone released by pituitary. It releases a hormone called thyroxine. It releases one more hormone that is called calcitonin, which helps in the calcium metabolism and the balance in your body. That is not in your syllabus. Thyroxine helps in the metabolic rate of your body. Okay, so in hyposecretion of thyroxine causes goiter, cretinism and maxedema, and hypersecretion exophthalmic goiter. Now when you talk about maxedema happens in adult, maxedema in adult, cretinism in children, but both are hyposecretion of thyroxine. Okay, and goiter is what? Enlargement of the thyroid gland because of hyposecretion of thyroxine. So all these three diseases, maxedema, cretinism and goiter, is because of hyposecretion of thyroxine, and hypersecretion of thyroxine is exophthalmic goiter. Eyes protrudes out, that is also called Graves' disease, right? Then another part of this chapter is your feedback mechanism. We'll understand with thyroxine. Now suppose your thyroid gland is activated by TSH, okay? So TSH is released, which goes and triggers thyroid to produce thyroxine. Now thyroxine level has reached to the level that is needed in the body. After that, what will happen? This thyroxine will go back to hypothalamus. Hypothalamus is the boss of pituitary, okay? It will go back to hypothalamus, which will ask pituitary not to release TSH anymore. So here the product itself is inhibiting the release of thyroxine. Thyroxine itself is inhibiting the release of thyroxine. This is called negative feedback. I hope it is clear. Yes, so let us move ahead to the last part of this unit, that is reproductive system. In this system, first of all, before talking about the reproductive system in human beings, you must be aware about what is sexual, what is asexual reproduction. When two parents are involved, the male and the female, and two gametes are formed, the male gamete and the female gamete, and they fuse to form a zygote. The process is called fertilization. And when two different parents of different gender are involved, that process is sexual reproduction. Asexual reproduction in plants, generally vegetative propagation, or by which only one parent is involved, that is called asexual reproduction. In sexual reproduction, the gametes are produced. In male, we have sperm, and in female, we have ovum. The structure of the sperm, that has to be very clear. You have a head, the sperm has got a head, a neck and the tail. Okay, drawing the diagram is very important. Here you have mitochondria present, right? You have a nucleus and then you have acrosomes. If you make all this, then you get marks for the drawing of the diagram. The nucleus, okay, the acrosome, mitochondria, that is there are many mitochondria present here, and the tail or the axial filament. This is the sperm, the male gamete. And in the female gamete, you have got round female gamete with the nucleus, okay, and it has got a vitelline membrane and the chorion cells, that is corona radiata, that is your different types of cells, corona cells are present around the ovum. This makes your female gamete. Okay, fusion of the male and the female gamete is called fertilization. Process of formation of zygote, that is fusion of the male and the female gamete to form zygote, is called fertilization. And the various organ systems responsible for the production of these gametes will be talking about. Like in male, you have got the male system. This is the urinary bladder. This is a part of the excretory system, not the part of the male reproductive system. Male reproductive system contains the different accessory glands, the ducts and the organ. Okay, so this is the male organ, which is called the penis, and here inside is the urethra. Urethra serves as a common passage for the sperm as well as for the urine. Both does not happen at the same time. This is called the sperm duct, which is also called the vas deferens. Okay, this is the testis, which helps in the formation of sperm. So here you have got different types of lobules, you can see that, which has got seminiferous tubules. Seminiferous tubules are the places where the sperm is formed, seminiferous tubules. And there are Leydig cells present here. Leydig cells or the interstitial cells are responsible for the production of the male hormone, which is called testosterone. Now the sperm produced here is not mature enough, so it goes to epididymis. This coiled structure is called epididymis. The sperm stays for some period of time and gets matured, develops tail. Okay, so it gains mobility. After the sperm matures, it travels through the sperm duct into the main organ to be ejaculated. Before that, it gets in contact with three glands. The first one is the seminal vesicle. The second one is the prostate gland, and the third one is the bulbourethral gland. Seminal vesicle, prostate gland and bulbourethral gland. The seminal vesicle releases a fluid which is called semen. Okay, so the sperm mixes with the semen. Prostate gland releases an alkaline substance which is required to neutralize the effect of acid present in the vagina or the female organ. And bulbourethral gland releases certain lubricants. Okay, this is the function. So now the sperm has mixed with the semen and all the alkaline secretions and the lubricants and move out from the penis through the urethra during the ejaculation through the ejaculatory duct. Is that okay? So this is all about your male reproductive system. In female reproductive system, you have a muscular organ called uterus. There is ovary. There are two ovaries. Each ovary functions in alternate months. There is fimbriae and this is fallopian tube. Fallopian tube. Fallopian tube is the site of fertilization. Uterus is the site of implantation. Okay, and ovary is the site of ovulation. So what happens, because of the impact of the follicle stimulating hormone, the primary follicle is formed here, which develops into the secondary follicle, tertiary follicle, and finally the matured follicle, that is called the Graafian follicle. Once the Graafian follicle is formed, the LH on the 14th day makes it release the ovum, which is collected by the fimbriae of the fallopian tube and it moves into the fallopian tube. After that, Graafian follicle changes into a yellow mass, which is called corpus luteum. Corpus luteum helps in the release of a hormone called progesterone, which is also called a pregnancy hormone. Okay, now what happens? Till the time it waits, corpus luteum waits for a certain period of time till the fertilization happens or it does not happen. So that is on 14th day it happens. On 14th day ovulation happens, and then for another say 5 to 6 days corpus luteum persists. If fertilization happens, it continues to stay for 9 months, keeps on releasing progesterone to maintain pregnancy. If it does not happen, it changes into corpus albicans and stops the production of progesterone. As the level of progesterone goes down, the menstrual cycle begins. So there is as I mentioned, menstrual cycle is a cycle of 28 days in females, which leads to the formation of the ovum, then also waits for it to get implanted and pregnancy to happen, or it does not happen. If fertilization happens, implantation occurs. If fertilization does not happen, menstrual cycle occurs. Okay, so onset of puberty in young females and the beginning of the menstrual cycle in the females is called menarche. Stoppage of the menstrual cycle in the old woman at 45 to 50 years is called menopause. So female remains fertile from menarche to menopause. Is that okay? So you know the site of fertilization, site of implantation, site of ovulation. The female hormone is estrogen and progesterone. What releases progesterone? Corpus luteum. What releases estrogen is the ovary itself releases estrogen. Now level of estrogen, follicle stimulating hormone, all hormones, so then regulates the menstrual cycle. Initially when the FSH is released, which helps in the formation of primary follicles, which increases the level of estrogen. Once the level of estrogen is increased, it goes to the pituitary to make it release LH, that is luteinizing hormone. Once luteinizing hormone is released on the 14th day, ovulation happens. Once ovulation happens, corpus luteum is formed, progesterone level is increased. As progesterone level is increased, the endometrium wall of the uterus remains thick and wait for the implantation to occur. If implantation does not occur, progesterone level drops and the shading of the endometrium wall begins. That is called the menstrual flow. So, menstrual cycle has got 4 phases, which is called M fall. Okay. What is the 4 phase? It starts with the bleeding. So, menstrual cycle, menstrual phase, that is 3 to 5 days of bleeding. Then you have the follicular phase, where the primary follicles get matured into secondary follicle and form Graafian follicle. After the Graafian follicle, that is maturation of follicle happens, there is ovulatory phase. On the 14th day, because of the increase of luteinizing hormone, ovulation happens. And after the ovulation, there is luteal phase, when the progesterone level increases and waits for a chance of fertilization to happen. If fertilization happens, luteal phase persists and the pregnancy happens and gestation period of. Let us talk about the baby growth. Here I have just erased the part that I have written earlier. Now this is the uterus. In the uterus there develops a membrane. Okay, that membrane is a kind of balloon like structure which has fluid inside, and the baby grows inside. Okay, and this is the baby. The baby is attached to placenta which is present. Okay, that placenta acts as a link between the mother's body and the baby's body, and this cord which connects the placenta to the baby is called the umbilical cord. The membrane which is just around the baby is called the amniotic membrane, and the fluid here is amniotic fluid. Amniotic fluid helps in absorption of any kind of mechanical shock and injury and protects the baby from it. At the same time, it also provides a pressure, equal pressure all around the baby for the proper development. Then outermost layer is the chorion layer. Okay, so now the baby develops. The placenta, you have understood. The allantois, there are finger-like projections from the placenta on both the side. One is with the uterus and the other one is the baby. The finger-like projection is called villi, the same as it was in the small intestine. Name is same, but function is also like the allantois. The finger, it is called allantois. That is the finger-like projection towards the embryo, and then the finger-like projection is also towards the uterus, that serves as a exchange medium between the mother's body and the baby's body. So this is then fertilization is clear. M fall, order is very important. Which hormone is responsible at which stage, that is important. Then development of the baby, the embryo, the fetus. What is the difference between embryo and the fetus? Fetus is when the embryo has got visible features like hand, leg, it appears human like. That is after 8th week. It is called fetus. An embryo is just the initial stage of mass of the cells. Fine. So the finally after 280 days of gestation period, the full grown baby is delivered. The process is called parturition. So this is all about reproductive system. Hope the concept is clear. Then there is the concept of twins. Okay, so how are twins formed? There is fraternal twins, then there are identical twins. So whenever there is a zygote formation taking place, and once after the formation of zygote, if the zygote splits, then you have the formation of identical twins. When you have 2 ovum produced by the female and it gets fertilized by 2 sperm, that is called fraternal twins. They may not be identical. Identical twins have to be of the same gender, and their genetics will also be similar. But non-identical twins are just like siblings, just they are born at the same time. They can be boy girl, boy girl, 2 boys, 2 girls, that can be there. But for identical twins, the gender has to be same, and they look also similar. Okay, there are cases of quadruplets, there are cases of 11 babies born at one time, and so many cases, right? So this is all about the human anatomy and physiology. Thank you for being so patient and good students. Thank you so much, children. Happy learning.